Hello, good morning, and welcome back to the channel. It's always beautiful to have you here with us. Today, we're going to talk about electrophoresis. Electrophoresis. Now, electrophoresis is a separation technique that is used in microbiology research, biotechnology, biochemistry, and many other various different types of research. And it's a separation technique that is used to separate biological molecules. Molecules could either be DNA, RNA, or protein molecules, and they are separated on the basis of two major principles. They are separated on the basis of their size, their mass, and the charge which they carry, their size or the charge which they carry. Now, many different molecules are not neutral. They may carry an overall positive charge or an overall negative charge. So this is a separation technique that uses both the mass and the charge that the molecule is carrying to separate these molecules and to be able to identify one molecule different from the other. So this is what electrophoresis is all about. When you talk about electrophoresis, you're talking about creating a system where you have an electric field, a charged electric field. You have this electric field, you create the electric field in a specialized tank. And when you create the electric field, you have the positive, then you have your electrodes, the negative electrode, which is the cathode, and the positive uh, electrode, which is the anode. You have these two electrodes, the cathode and the anode, and then you create an electric charge in. So you create an electric field with an electric charge, and you have these electrodes, and then you have wells. We call them sample wells, where you load your samples into. When you load your samples in this well, you put on your your source of electricity, electric current. You create this field, and then you know what happens is you also have a medium through which the medium acts like a what do you call it, a sieve. Medium acts as a sieve because the particles are going to go through this medium. So the particles, the ones that are lighter and depending on their charge, will move faster, obviously, than the ones that are slower and heavier. Remember that it's simply a separation technique. So depending on what medium they are going to put in that tank, that was also determine the kind of name that you give to the electrophoresis, because there are different types of electrophoresis. So sometimes we just say gel electrophoresis. What we're actually saying is agarose gel electrophoresis, and in that instance, agarose is used as the medium through which these molecules are going to pass through for them to be separated. You can also have, apart from agarose, you can also have other media. Sorry, you can also have other media that you use to for this separation. You can have polyacrylamide as a media that you use for this differentiation. So you call it polyacrylamide gel electrophoresis, which is the page, polyacrylamide gel electrophoresis. Then you can also have SDS, sodium deodecyl polyacrylamide gel electrophoresis. You call that one SDSS page electrophoresis. So the idea is that the electrophoresis is a separation technique and you use it to separate RNA, DNA, and then you use it for even some protein analysis. And so it's important and it's in use today for very, very, very different methods. Also, you have paper electrophoresis. So you can actually use a Wattman's filter paper as the medium. So you create an electric field, you put the filter paper there, and it is used as a medium. You load your samples and you put on the current and you use it as a medium, and you watch the separation of different molecules using that type of technique. You also have a what we call the capillary electrophoresis, which uses capillary tubes for the electrophoresis to take place. So it's important that we understand what electrophoresis is and why it is being used, its application. And this would help us in our knowledge of science. At the end of the day, it's important when you do PCR, when you do different kinds of biological techniques, it's important that you're able to separate molecules on the basis of their size and the charge which they carry. Because when you have this process taking place, of course, the positively charged molecules will gravitate or move towards the negative electrode, which is the cathode, while the positively charged ones will move towards negative. The positive molecules will move towards the negative electrode, which is the cathode, and the negative molecules will move towards the positive electrode, which is the cathode. And at the end of the day, you see the separation of the molecules, and then you can go further in your analysis depending on what you are doing to either maybe run a PCR or do whatever it is you want to do to be able to identify these organisms or see what it is that you are searching out for. Thank you for listening. That's just a short discuss on electrophoresis. It's a separation technique and it is useful for us in science. Do have a wonderful day.